The following program is brought to you by Caltech.
So I want to first talk about the start of the era, which really started back in 1967 when Max Delbruck popped into Carver's office, and in my best imitation of a male German voice, he asked Carver the following question. I hear that nerve channels are like transistors. Is that true? And anyway, Carver at the time did not know who Max was, but after reading the papers that he got from Max, he decided that he would like to do some device physics on the artificial bilipid membranes. And so Carver then contacted Paul Mueller, who was at the time a well-known biophysicist who has been working on bilipid membranes in Pennsylvania, and ordered Paul Mueller to come out to Caltech and to work with Carver and to teach him all about membrane biophysics and how to build artificial membranes. And this work continued for a while with Moshe Eisenberg and Jim Hall until the 1970s when Carver returned to his research in computer engineering.
Jim's here, and he'll talk a little bit later.
Yes, right. And so, however, the interactions in this period started Carver thinking about how device physics can be used to model biology. And so the next thing that happened was in the early 1980s, there was a physics of computation class that was put together by John Hopfield and Carver Mead and Richard Feynman. And apparently the lectures were very interesting. The material was so exciting that John Hopfield, along with David Van Essen and also John Allman, started the Computation and Neurosystems program in 1986. And Christoph Koch was one of the first faculty that was hired into this program. And the physics of computation class then split into three. So there was a class taught by Richard Feynman, another taught by John Hopfield, and Carver taught the one about neuromorphic systems, which also became known as CNS 182. And it was a course about learning about subthreshold analog circuits, and also about the building blocks of different parts of the nervous system. And there was a class that I took, and it was a very long class. In fact, we teach this class now to our students in the Institute of Neuroinformatics in Switzerland, and the students will complain about how long these labs are and that we should really cut back down on the hours. And so we really had to cut back like about 60% of the class material. And so I want to talk a little bit now about the lineage of the people that were in this neuromorphic times in Carver's lab. And so the person that I really want to talk about first is Misha Mahowal, because Misha was a key person that was instrumental in the growth of the neuromorphic field. She was an undergraduate biologist, and she had taken the physics of computational class, and she was introduced to Carver by David Feinstein, who was a TA in the class. And together with Carver, they came up with the first silicon retina, and then she worked on the silicon neuron and also the stereoscopic system. And during the next few years, it was really a golden era for Carver's lab, because the students could really pick any part of the nervous system and work on it because nobody else was doing this. And I also want to mention the visitors that came into lab. So there was Dick Lyon, Andreas Andreo, and Basen Lander. And two other people I want to talk about. So one of them is Eric Vitos, who was in Switzerland, and he was credited with saving the Swiss watch industry because he was really an expert in analog subthreshold circuits. And when he saw Carver's work and also that of the students, he decided he was going to get into the same field, and he started his own group in Switzerland where the students were also working on similar systems. And then the other person I want to mention is Rodney Douglas, who was a frequent visitor with Christoph Koch, and during this time had also started out some collaborations with Misha Mahowal, and I'll talk a bit about their work later. And so finally, during this time, there were also students from different eras. So for example, Mass and John Lemanchek, Sylvie and Marianne, and also students that were shared with different faculty like John Hopfield and also Christoph Koch. And finally, you get the set of 6 over here. And so this is the group of 6 students that basically ended the neuromorphic era in Carver's lab. And so this is a typical Carver Lab meeting. By the way, Carver Lab is Carver Lab. And the students would take turns bringing food. So Carver was very good about making sure that people were full while they were discussing science and technology. And so this is an example of kind of the intense discussions that happened. after dinner. After dinner, is that right? Yeah, and so you can see Mass and David Gillespie, Basson, and then Ron Benson and the three Johns. and out of all the work that came out during this time was this lovely book that Carver wrote about analog VLSI and neural systems, where the chapters covered different projects in the lab. And so it led to a whole bunch of books about neuromorphic engineering, as you can see over the years. And so the latest one is one that's coming out, written by some of us at the Institute of Neuroinformatics. And so I want to talk about 4 different areas. So the first one is of course, about the retina. And so the silicon retina was really the first neuromorphic system that was developed in the lab by Carver and Misha. And so you can see an example output from this particular retina. And so this retina implemented the center surround properties of the biological retina. And so basically consisted of a log photoreceptor where the output now drives a resistive grid, and this resistive grid computes the spatial temporal average of the surroundings. And by taking the difference between the output of the photoreceptor and also that of the spatial temporal average, you can get the edges, if you can see, of the object. And so even though this retina had a scan output, Misha and also Mass Cibolati had worked on a spiking version of the retina where the outputs were really asynchronous spikes, and they carried precise timing information of events in the world. And this is the kind of different kind of output that Carver would usually say that it's an output where time represents itself. So you really did not have to have a clock in the system. So different students have gone on and started different labs, different companies like Tanner and Foveon and Synaptics. And the retina basically got better with new designs by Corbena Boerhan, who also worked on new implementations of what we call the AER protocol, the Address Event Representation Protocol. There were new motion chips that came out where we're now hoping to put them on quadcopters. And then I'm going to talk about a specific system here so that you can see what the outputs of such a retina looks like nowadays. So this is the dynamic vision sensor that was done by Toby Delbrook. And so it's a lovely design that really tries to reduce the mismatch that you see in the original retina. And the cells only put out spikes that tell you something about the change in the temporal contrast at the local pixel, right? And so what I'm going to show you now is a video from the latest chip, which is called the Dynamic and Active Vision Image Sensor, which puts up frames and also this DVS output so that you can see how sparse these outputs are that are being sent. Okay, so what you see here are images from the sensor. It's 240 by 180, and so this is like taking a picture over time where you can see even if most of the scene is not moving, you're taking a picture and basically sending a lot of data across to your processor. But if you now look at the DVS output, where you see the red and green lines, and so those represent the on and off temporal contrast of the image. And so what you send across now are these really sparse outputs, because the only thing that's really moving in the picture is that of the doing cartwheels on the lawn. And so it really tells you that your retina is sending very sparse output up to the brain. And so it's just a lovely idea of what the advantages that neuromorphic system can bring to the processing world. And the other sensor that I want to talk about, another key neuromorphic sensor, is the Cochlea. And so together with Dick Lai and Carver had come up with the first silicon cochlea, which consists of a cascaded filter bank. And so here you see the output of one of the filters. And Carver was very fond of positive feedback. So in many of the circuits, you'll see positive feedback. And so here you can see the positive feedback in the filter. And this is something that later on Lloyd Watts will talk more about. And this work led to different kinds of chips. For example, this is the Sharma chip, which was named after Shiab Sharma, who came and gave a talk about a model of sound localization. Also led to Lloyd's company called Audience. Corbena Boerhan did a current mode version of the Cochlea along with a new automatic gain control mechanism. Rahul Sapeshka took the circuit and did applications for cochlear implants and also RF cochleas. And I, together with Andre Van Schaik from Eric Vitos' lab, designed a binaural cochlea that would put out AER spikes. And so the third area that I want to talk about is the silicon neuron. So this is a circuit that Carver came up with called the axon Hillock circuit. And it's a circuit that we still use nowadays in many of our chips because it's such a simple circuit. And it also, again, uses positive feedback. Right? So the neuron, the circuit implements the integrant fire mechanism of the neuron. And so what you see here is current charging up the membrane of the neuron, which is modeled by a capacitor. And whenever the voltage crosses threshold, then an additional output is generated. And by this positive feedback, you basically ensure that the output is all or nothing signal. And so this circuit led to a more biological realistic circuit that was done by Misha Mahawa and Ronnie Douglas, appeared on the Nature cover, and also then led to more and more chips that had more and more neurons. The latest is a neurogrid system by Paul Bennett Bohan that has 1 million neurons. And just today in Science was the chip by IBM called True North, where 3 of Corbena's ex-students were instrumental in designing the chip. So you can see the influence of Carver is just amazing. From his original circuit, it's just amazing till today. And also, there are a lot of interesting things that are going on. So one of them is the kind of processing that you now have to carry out on the chips. So Terry Sanowski, along with Toby, had come up with a possible idea of how you can transfer information from your inputs into these multi-neuron chips that don't have a clock. And one way is by using the temporal synchrony of your inputs. And then Rahul Sapeshkar has taken it even further by combining 2 different fields, electronics and cell biology, into a new area called cytomorphics. The fourth area I'm going to talk about is about adaptation. So one thing you notice is that there's mismatch in the silicon retina of Carver and Misha's on the Scientific American article. And so Carver decided to use the floating gate technology to reduce the mismatch that you see in the array. And so this is an example where you would have to erase the charge on the floating gate using UV. And together with Brad Minch and Chris Iorio and Jen Hassler, they came up with new electrical mechanisms, which include tunneling injection, to move charge on and off the floating gate. And from this development, the team designed things such as the learning synaptic array, which Chris will talk more about. New companies started, Impinj by Chris, Getronics by Jen Hassler, and also a set of field programmable analog array chips where the floating gate technology was used as a way of programming interesting analog circuits. And finally, on this slide, I wanted to talk about the legacy of Carver to this whole neuromorphic field. And so you can see that the field that he started led to basically new labs that came from his ex-students and to now the students' lab. So it's really like an exponential influence on the world. Also led to the Institute of Neuroinformatics, where I'm currently at. We're now a big presence in the conference ISCAS, which is an IEEE Circuits conference. New conferences were started by visitors in the lab. For example, BioCast, which was started by Boston. And then the Telluride Neuromorphic Cognition Engineering Workshop, which was started by Christoph Koch and Terry Sanowski, just celebrated their 20th year. We missed you there, Carver. And then there's also now a sister workshop over in Europe, started by Ronnie Douglas and Giacomo Indiviri, called the Capocaja Workshop. You'll see that all these workshops are in kind of these areas where you really can easily get into town and do fun stuff, because the idea is to kind of lock you together and to really work on things together. So it's really a workshop. And then finally, I just want to talk about how neuromorphic has changed from when I first was a student in Carver's lab. So really now anything that's bio-inspired, people will call it neuromorphic. Address event representation, or AER, is now the signal protocol that's used in many neuromorphic chips. Mismatch has become less of a problem because we understand how to do the circuit design better. In fact, it's almost a feature now in the new networks that people are considering. Computation is still a challenge for the chips that are currently being constructed. Nano devices that hold memory, like memristors, are also now considered neuromorphic. And then finally, logic is becoming interesting again as technology scales down, especially probabilistic forms of logic. So it's really amazing, the kind of the change in the field. And then finally, I want to leave with this picture, which was really the group of students that were in the lab before the Neuromorphic Era ended in Carver's lab. And what's that? You can probably recognize some of the people there. At least you can try and pinpoint who they could be. And really, I want to thank you, Carver, for your inspiration, your exemplar work habits. And you seem to design more chips than anybody else, including your own students. And also being part of my personal life, how you came to our wedding and spoke at our wedding. The multiple dinners that we have at 1510 Oakdale, which is where we're going to have the party tonight. And finally, I guess we will all continue, as you used to say, is to listen to the silicon. And happy birthday, Carver.
I have to do a little rebuttal. That neuron circuit I didn't invent. I don't remember who did, but I did. So,
I guess we always
does anybody here remember who invented that circuit?
say it was your
You
circuit.
don't even remember that you invented it.
It's your circuit. It's an axon hillock circuit.
The other thing that everybody here should know, of course, people like Casey and Kelly never wore their own heart, so you never know that they have gone out and done great things. Oh, it's not here. Let me, does that work better?
No, it sounds.
Oh, okay. I just put the switch the wrong way, that's all. You know, you get a sign wrong now and again. That picture right there, you probably noticed that, you know, there's Kwabena and Rahul and Brad Minch, I guess, didn't make it. Is that right? Oh, there's Brad. Okay, great. And Dick Lyon you recognize, and then right next to Dick is Candace Schmidt, who was working in the office with Donna. So you might ask, where is Donna?
Oh, gosh.
Well, it turned out, you know, in her younger years, Donna had worked as a model. She, you know, but she was too shy to get in the picture for this one. So we missed Donna. And there's Sanjoy and Jim Campbell, I guess, is here. Yes. Jim, there's Jim right back there, yeah. And Jen Hassler and Lena, who is now a professor in
Lund.
on the West Coast.
She's at Chalmers.
Oh, Chalmers.
Chalmers,
that's right.
yeah, she's at Chalmers, yeah. And doing well there. And so basically everybody on this slide has gone off and done, oh, and Andreas isn't here, is he?
No.
Andreas is now
Johns Hopkins.
Johns Hopkins, yeah. He's been at Johns Hopkins for years.
Yeah.
Didn't we get Kwabena from?
Yeah.
Yeah, isn't that, you came from Andreas's lab?
Andreas who pointed me in the right direction.
Yeah. that was great. So we've got these friends. The other thing that, of course, Shichi would never tell you is that since she left here, she sort of taken on herself the task of keeping everybody in the community collaborating with each other instead of fighting tooth and nail. And that takes a very fine set of skills. And Shichi has been sort of the worldwide magnet for getting these conferences, the sessions organized, and getting people together to contribute to the books and things. And so I want to thank Shichi for all her contribution to the several generations we're seeing here. So
Thanks, Carver.
All right, great. I forgot to mention a couple of great stories after Shichi's talk. So, well, being as disorganized as usual, we'll do it now. Shichi mentioned that Max had this habit of, when he wanted to do something, he would call up somebody and order them to do whatever he wanted. And he was very persuasive that way. And it was a time when the...
As kids, no.
Yeah. That was just at the time when artificial lipid bilayers were first being used as a model for what various molecules might do in nerve membranes. And so Max and I had gotten corresponding and were looking at some of the literature together on that subject. And so Max called up Paul Mueller and basically ordered him to send us some of the stuff and to come out and teach us how to do that. So that's how I met Paul. Paul came out from Penn and spent a week with us, and we started a little effort. And it turned out that we were sort of fumbling around, and we got this message from this, but long story, but we got this message from this fellow in the service that said he could get out a year early if he had an academic position. And he had been working on membranes of various sorts before he'd gotten drafted. And so we pulled some strings and hired Jim Hall to come here for a year. And then there was a grad student by the name of Moshe Eisenberg, and the two of them worked in the sub-basement of steel, because down in the very bottom of one of the rooms there was a pillar that was independently supported down in the earth, so it was supposed to be a vibration-free thing, and these lipid membranes were very sensitive to vibration. So that was what Max called our subterranean group. So Jim is here to tell us a little about that era, which was actually, we made some progress.
So Carver, your memory is really good. Do I need a
Come over
microphone?
here to the microphone. perhaps.
Do I need a microphone?
Yes.
No. Microphone, raise your hand. You're not gonna like it. So this was Carver. Your memory is really good because you remember a lot of things that didn't happen. And actually, I got out of the service by teaching high school in San Bernardino.
Oh, okay.
And I came over to Max because he had had a conversation with my thesis advisor. So I said, Max, I want to work on lipid bilayers. And he said, that is not my project. And Toby, who stole the umlauts?
What's?
There's no umlaut on the U.
Well, we don't like.
Okay, anyway. So Max said, you have to go and talk to Carver Mead. So I went over and found Carver. I don't know, you were moving, with 2/3 speed of light or something up on the 3rd floor of steel. So I went over and talked to you and said, I want to work on lipid bilayers, and here's what I've been doing. And you said, oh, that'd be great. You'll do an NIH fellowship. You'll come back. We'll work, and we got a lab and everything. And I said, well, what if I don't get the fellowship? Will there still be a lab? And you said, yes, there'll be a lab. It'll be down in the basement. It's right close to all the electronics for the undergraduate lab, so we got plenty of stuff to build things. And not only that, it's deep, no vibrations, and no electrical interference, because we knew we were going to have to build picoamp sensitive current measurements. Well, it turned out that this room was great. We built all this apparatus. We got everything going. And then we discovered that on the other side of the wall of our building, our little room, was the substation for the entire building. We learned a lot about electrical shielding. But we had a lot of fun. How many of you guys have heard of the word ion channel? How many have not? Okay, the ones who have not, you are full of them. And in fact, the analog.
The ones that heard it also full of them?
Yes, they are too, but in a different way. So in the mush analog digital misunderstood computer that is between everybody's ears, they're ion channels, and they are what make everything work. At the time that we started the subterranean membrane group with Moshe, Carver, and Max, the number of known ion channels was possible to count on 2 fingers. How many are there now, Scott? 25, 30, 50, 200? I don't know. But they're really, and we were down there in the sub-basement among the first people to see the actual signature of a single molecule of ion channels. So with roughly $50 worth of stolen op amps, we could see the fluctuations of a single molecule, the current jumping up and jumping down. And I have to admit, that was the first time I actually understood the kinetic theory of gases, because we had to do all this stuff. Can I take 2 more minutes to tell a story on Max?
Sure.
So what we had to do was we had to figure out that the probability of the channel opening described the IV curve that we saw, the very voltage-dependent IV curve. So we did all of these great statistics. It turned out we did all these measurements. And one of the cool things about Carver was there was nothing that you needed to do that he couldn't find a path for you to do it. And we had to measure probability distributions, and we didn't have a machine that would do it. So do you remember this, Carver?
Yeah,
we modified this Princeton applied research waveform adductor by putting all this stuff in, and we got these great probability curves, and the data all came out perfectly. And I didn't know anything about probability or statistics, and Carver said, "You go to the library, read Fowler, it's the best book." So I went to the library. Caltech had a library then. And a bookstore. So I read this stuff and did all these great statistics, and I had 2 pages worth of proofs of why this probability theory worked and everything. And we wrote the paper up, Moshe and I wrote the paper, and we took it to Max. And Max read the paper, and he called me over to the house that we're going to tonight. And I sat down with him, and he said, I don't remember the name of the fallacy that I was accused of having made. but here I am arguing statistics with Mr. Luria-Delbruck fluctuation experiment. And this is for those of you who are not biologists, this is an incredibly important biological paper. And I said, "No, Max, look, I proved this thing. I'll give you I've got 2 pages worth of stuff." He said, "I'm not going to read that." He said, "Go talk to Ed Lipson. He will explain it to you." Ed Lipson was a postdoc of Max's. And so I went up to Max and said, "Ed, Max says this is all wrong. I can't get the thing done." And he said, he said, "Well, explain it to me." So I said, "Here's the way it works." He said, "Max is completely wrong. You're absolutely right." He said, "Now we have to figure out a way to explain it to Max." And Ed came up with about 2 lines of really non-rigorous but convincing mathematical argument that made it into the paper. So that was it. Anyway, that was fun. Sorry?
What was the ion channel?
That was Alan
This was
Matheson.
Alan Matheson.
Yeah.
It was a highly voltage-dependent ion channel.
Yeah.
And then I went on and did basically cytoreactive mutagenesis and all that.
And what was important, can you hear me? Am I here? Am I on? Yeah. What was important about that work was that the papers that had been published up to that time had assumed that the exponential current voltage characteristic was a function of each channel individually. And what that work showed was that that was a function of population, not of, and it was in the probability of opening and closing that. And of course, nobody ever quotes that paper because it was about Alan Matheson. And you know the way biology is, they always look at what the preparation is, not what the scientific finding is. So that was. So if we could get the other slide. It turns out that Jim's from Riverside, and Scott, where'd Scott go? Come on, Scott, come on up. Scott is also from Riverside, or was.
No, from Irvine.
Irvine.
Sorry, that was down that way someplace.
Yeah,
you know. Sorry.
One of those Orange
And,
County places, except Riverside.
Scott has another story that's illustrated in the upper right corner here, which he's going to tell us.
Well, I just commented at the coffee break. I'm Scott Fraser. I was here for 2 decades and lucky enough to be in Carver's Wednesday lunch group with Dick Lyon for some of the most wide-ranging conversations we could have ever had. And one of the very fun things about that is, as a result, I got sucked into several of the thesis committees for Carver students, Toby being one of them. And that slide up there, that deserves more comment, including the movie that we saw earlier. So Toby's thesis defense has, in my mind, and every one of my students' minds, even though they weren't there, gone down in the sort of annals of best thesis defenses ever. Because a part of doing the Retina and the gain control circuit, and Toby will probably either kill me or correct me if I get it wrong, part of the challenge was to build a circuit that would work in a huge range of illumination. I mean, just as we can see and hear, and we saw out at the break, and the illumination is dramatically different. And so Toby's performance at the thesis defense was to set up his artificial retina, looking at the scene of the room, and show that it could work as he walked around. As I remember, he did not do somersaults or handstands or anything. And then he dimmed the rooms down to about this level. After the lights were up full intensity, dimmed it, showed that it could still work, turned down the lights even more, showed that it could still work. And being the hot dog grad student that he was, he eventually crossed himself, extinguished the lights, and lit a birthday cake candle. Am I getting this right? Yeah.
Yeah.
Okay, it's gotten better in my mind than it probably ever was.
No, that's
and then he showed that it still worked. And he could have just walked out of the room then and we would have passed him. He had to go through the rest of the defense. But it was just an amazing demonstration how much better it was. Those of us that were building microscopes and microscope cameras at the time both loved and hated that work because we couldn't get anywhere near to that dynamic range. So, Toby, that picture tells a lot of stories.
Right.